Today we're going to study some of the methods that are used for protein isolation and protein detection. Our objectives are as follows. Number one, we're going to look at the component for lysis buffers such as detergents, chelators, and inhibitors. And then we're going to look at where we go from isolating the crude extract that contains the proteins, and then how we're separating proteins based on two natures, charge and size. And then finally, we're going to finish up the lecture by looking at two protein detection methods, such as Western blot and immunofluorescence. Let's begin with isolation. When you lyse cells, you use three things in your buffer: detergent, chelators, and protease inhibitors. Detergent is to lyse the membrane. Chelators are to remove all the metal ions that might be necessary to activate proteins. And protease inhibitors are used to inhibit proteases that to prevent them from chewing up the proteins of interest. And then separation of proteins, you can separate them based on charge and based on size. Let's look at cell lysis. When you lyse the cell, you have membrane and you have transmembrane proteins. And then you have proteins that are in the organelles or they are in the cytosol. Now, the detergent that you choose can either generate proteins that are intact, non-denatured proteins, or denatured proteins. So if you use denaturing detergents such as SDS, you will isolate proteins that are denatured. If you use Triton X or CHAPS that are non-denaturing detergent, you will isolate proteins that are where the three-dimensional structure is intact. And you can dialyze away the detergent and obtain protein activity or enzyme activity. What allows us to use detergent to destroy the lipid bilayer? Well, it is inherent in the nature of phospholipids. Phospholipids consist of a glycerol backbone and then two fatty acids on carbon number 1 and carbon number 2, and then a phosphate group on carbon number 3. And when you put them together, they have the following. They have a non-polar head, tail right here and a polar head, so that when you draw them, you have a polar head and a non-polar tail. Again, polar head, non-polar tail right here. And you have fatty acids on carbon number 1 and 2, and then you have phosphate groups such as phosphatidylethanolamine, phosphatidylserine, and phosphatidylcholine on carbon number 3 to give it the polar head. Now, when you put phospholipids in aqueous solution, they will associate to form micelle or lipid bilayer, and that is that the polar heads will be oriented into the polar environment. The non-polar head will be oriented out of the polar environment, and they will be associated with one another to sequester the polar molecules away from them. There's a process called saponification that allows us to use detergent to lyse the membrane. Saponification is a process where you get formation of salt of detergent right here, so that when you put them, and this is an example, salt of detergent is called soap. You get fatty acid and potassium or sodium associated with it. When you put the salt or the soaps into an aqueous solution, they will form a micelle. And so if you were to add the micelle of soap with a membrane or a grease droplet, they will merge with one another right here. You got the grease droplet right here, and then you've got the soap right here, or the salt of fatty acid, and when you combine them together, they will form this combined structure right here. And so if we were to assume that this is the membrane right here, and this is the detergent, detergent will indeed destroy the membrane structure. Again, as we said, detergent can be denaturing and non-denaturing. Detergent can be ionic or non-ionic, and this is one example. The formation of the polar head is ion. The polar head here is non-ionic. There's no charge here, but there's a charge here. That's the difference between ionic and non-ionic detergent. I'm going to fast forward from here. So if you were to mix the detergent and a cell or the membrane, here's what happens. You've got the detergent that will form and associate with the proteins right here. And if it is non-denaturing detergent, it's going to surround the protein right here, and it will not cause any denaturation. And then the detergent molecules will combine with the membrane lipid bilayer right here to destroy the integrity of the lipid bilayer. So when you add the detergent, it will tease out the protein and it will destroy the lipid bilayer right here. And that's how detergents work. Here is again another example, and this is a better view. You have the membrane bilayer, lipid bilayer right here with all the membrane proteins right here. When you add the detergent will separate out the proteins, and this is non-denaturing detergent, and then it will also destroy or break up the lipid bilayer right here. Now, if you were to, you can purify the proteins, and you can dialyze away the detergent right here, but if you were to combine the proteins that you just isolated from the membrane with phospholipids right here in a polar environment, and you dialyze away the detergent, you will get formation of the lipid bilayer and then the transmembrane proteins right here. Okay, so this is how detergents work by lysing the cell membrane. Now, let's talk about denaturing detergent. Denaturing detergent, when you use denaturing detergent, all of the proteins are going to be denatured. One of the most often used denaturing detergent is STS. STS denatures all the proteins. And you can use denaturing detergent when you just want to isolate the protein, the total protein, and you want to detect the presence or absence of a protein or to what extent it exists in the cell, but you really don't care about the activity. And that's when you use denaturing detergent. Non-denaturing detergent is that proteins remain intact. And one of the most often used non-denaturing detergent is Triton-X 100. And this is done if you want to isolate proteins and you want to measure the protein or the enzyme activities of these proteins. So you want the three-dimensional structure to be intact, and so you use the non-denaturing detergent. And then before you do the assay of enzyme activity, you normally dialyze away the detergent molecules right here. Okay. Now, once you get the crude extract, you can begin to separate the proteins of interest. The first way that we can separate the protein is by separating them by charge. And there are three methods that we can use to separate the proteins based on their charges. The first one is electrophoresis. Second one is isoelectric focusing. And the third one is ion exchange chromatography. And we're going to go through all three of these. The second way that you can isolate protein or separate protein is by separating them based on their size. And you can do that by centrifugation, differential centrifugation. You can do that by molecular exclusion chromatography. And the final one that we're going to talk about is polyacrylamide gel electrophoresis. Let's begin with separating proteins based on their charges. Now, one of the most often used method of separating proteins based on their charges is polyacrylamide gel electrophoresis. This is an acrylamide molecule right here, and it is usually made up in a solution, and it is mixed with bis-acrylamide, which is a cross-linker, to form this polymer right here. Okay, and so you go from individual molecules to polymer right here that can be made into a gel like a jello right here. Okay, and so the way you do it is you normally pour a certain percentage of the gel, and it's sort of like the agarose that you may have worked with. You put combs in here, and so to separate the proteins, you basically load the crude extract into each one of these wells. You subject this to an electric field, and the proteins begin to migrate from one pole to the next. All right. Now remember that in nature you're isolating these proteins based on based on their charges, and you're separating them based on their charges. So if you were to isolate the crude extract and you do it by non-denaturing gel, each one of those proteins is going to have an inherent charge, net charge. Some are going to be positively charged, some are going to be negatively charged, and some will be zero charge. And so they're going to migrate from the negative pole to the positive pole based on the net charge that they have on them. Okay, and when you stain the gel after running it, you will see an array of proteins right here based on their migration on the using the net charge that is on each one of them. The second method is called isoelectric focusing. Now, isoelectric focusing is separating the proteins based on their isoelectric point. Now, isoelectric point is the pH at which the net charge on a protein is equal to 0. And so what you normally do is you generate a pH gradient that may go from 0 to 14. Okay, 0 to 14. And then you load that, whether it be a membrane or a gel. So you load the proteins, let's say you load the proteins in the center, or you load it at one of the poles right here. And when you begin to subject this to an electric field, you generate an electric current, the proteins are going to be separated, and when they migrate to the point where the pH is equivalent to the isoelectric pH, or they're going to migrate based on the net charge until they get a pH where the net charge on each one of them is equal to 0. And so the ones that have lower pKa, they're going to migrate to a point at the pH where the low pH where the net charge on that protein is equivalent to 0. The basic proteins will not migrate until will not stop migrating until you get to the near neutral, the higher pHs, where the net charge is going to be equivalent to 0. And so again, the proteins are going to be migrating in an electric field, and they're going to seek out that pH at which the net charge on that protein is going to be equivalent to 0. And the final one is called ion exchange chromatography. And so you have an extract, crude extract of protein that proteins have a variety of net charges on them. Okay, you can have a column, set up a column that consists of beads right here that are coated with either positively charged or negatively charged groups right here. This one just happens to be a positively charged group. Now, I need to explain a terminology here. If you have a column, the matrix right here has associated with it positively charged group, you know that this positively charged group will accept negatively charged group. And so this is called an anion exchange chromatography. However, if you have a matrix that has negatively charged group attached to it, it's going to be accepting positively charged group, and this is called cation exchange chromatography. Okay, so let's look at this right here, for instance. So you have the matrix, and then you have a matrix, and then you have the positively charged group that are attached to it. If you run the protein samples along here, every protein that is negatively charged will be bound to this. Okay, so let's say that you understand the charge on the protein of interest that you have. You're going to run the buffer with the pH to make sure that at that pH your protein is negatively charged so that it can bind to this. Now, you wash it and all the other proteins are now falling off of the column. Your protein and maybe other proteins that are negatively charged will be bound to that column. Now, how do you elute those proteins? Well, the way you elute it is you just begin to change the pH of the wash buffer. You change the pH of the wash buffer so that it can get to the point where the pH of the wash buffer is going to be, it's going to convert your protein from negatively charged to zero charge. And once you wash that off with a pH where it's going to convert your protein from negatively charged, that is bound to the matrix, to a zero charge, that protein of interest is going to be washed off. Okay? There's another way of eluting that protein from the column, and that is, look at this picture. This column is positively charged, and it binds your protein which is negatively charged. All you have to do is compete your negatively charged protein with negatively charged ions, such as chloride ions. So you add in sodium chloride, and the chloride, if it exceeds at the ratio of 1 to 100 or 1 to 1000, those chloride ions will compete for this positive charge, and they will win the competition because they're more numerous than your protein. And so if you wash it with increasing concentration of sodium chloride or ionic strength of sodium chloride, it will elute your protein off of the column. In the application practice or in the team application problem, we're going to look at this. I'll give you some problems so you can practice. And so let's begin to separate the protein based on their charges. One of the ways that you can separate proteins based on their sizes is by differential centrifugation. And so you go with a lower speed to spun out the heavier protein, and then you're just increasing the speed or centrifugal force right here. The lower speed will pellet the heavier proteins, and the lighter proteins will be in solution. That's how you separate them. And you just kind of step it up. You increase the centrifugal force, and you continue to increase it, where it gets to the point where the only the really light proteins will be pelleted at the end, whereas the larger proteins will begin to pellet at very low centrifugal force. The second one is molecular exclusion chromatography, and that is that you use a matrix that has pores in it. Now, you can order these matrices right here with different size pores, and depending on the size of the protein that you're looking for, or the molecule that you're looking for, you can actually order a pore size that will, where your protein, because of its size, can spend a lot of its time interacting in the pores, within the pores. The larger molecules will be flushed away from the matrix because they're too large. They don't interact with the pores in the matrix, and you can just wash them off and wash them off very quickly. Okay, your molecule is going to interact with the pore, and you can begin to elute it after you wash off the larger molecules. Now, the smaller the proteins, the longer the interaction it's going to have with the pores within the matrix. And so you can actually separate proteins based on that. The ones that interact with the pores the most will stay the longest in the matrix. The one that interacts the least will not stay very long in the matrix. The one that don't interact at all will move on much quicker than the other proteins. And the final one is polyacrylamide gel electrophoresis. And the way that we separate proteins by polyacrylamide gel electrophoresis, the first one that we looked at was separating them based on their charge. Here we're separating them based on their size. So what you usually do is, if you were to use a denaturing detergent, that detergent is going to destroy the three-dimensional structure of the protein. Now, there are three things that are in play. There is the shape of the protein that will affect its movement. There is the net charge of the protein that will affect its movement. And then there's the molecular weight of the protein that will affect its movement. If you use a denaturing agent such as STS, or sodium dodecyl sulfate, what you do is that the STS is going to denature every protein completely, so that you're going to linearize these proteins, and not only linearize them, but you're going to attach the STS molecule to the protein where that protein is now negatively charged. And so in essence, what you have done is this, that you have destroyed the effect of shape, you have destroyed the effect of charge, and the only thing that they are separated based on is size. And so that's how you can separate these proteins. And so when you run the gel, of course, they're going to move from negative to positive, and the smaller one will move faster than the larger ones. Protein detection. Protein detection is based on the following principle, and that is that you have the antigen, which is a protein. It can interact with an antibody that is made against that protein or against that antigen. Now, after you interact that antibody with its antigen, you can also interact another antibody, and that antibody has an enzyme conjugated to it. That antibody is specific for the antibody against the antigen. And that antibody, so the antibody against the antigen is called primary antibody. The antibody against the antibody for the antigen is called secondary antibody. So you can interact the primary antibody with the antigen, wash it, and then you interact the secondary antibody, which has an enzyme conjugated to it, with the antibody and the antigen complex right here. And then when you add in the substrate for that enzyme that is on the secondary antibody, you can visualize the presence of a band where the interaction takes place. And this is basically the principle of protein detection. Okay? So protein detection is done by Western blot, and Western blots has the following steps, and that proteins are separated by gel electrophoresis on acrylamide gel. They are transferred from here to a membrane. Again, you're just making a replica of the gel, a membrane, because you cannot hybridize an antibody with this gel, but you can with the membrane. And you can do this by electrotransfer. Again, they're moving from negative to positive charge. Then you hybridize the membrane with the primary antibody, and then you wash, and then you hybridize the membrane with the secondary antibody that is conjugated to an enzyme. So you got acrylamide gel, transferred to a membrane, hybridized with primary antibody, then you hybridize with secondary antibody, and then you add the substrate that's going to develop a color on the membrane, or you can expose it to a film, and you get, out of these thousands of protein bands in the lane, you can get 1, 2, or 3 at the most, depending on how specific your antibody is. The second one is you can actually detect protein directly inside cells. And so let's say I want to treat the cells and I want to see the effect of my treatment on the sodium calcium exchanger protein, which is a membrane protein. So what I do is after treatment, I fix cells in paraformaldehyde to arrest the methods or the processes that are taking place in the cells. And then I poke holes in the membrane by permeabilizing it with Triton X 100, a very small percentage, about 0.1%. And then I do it exactly the same way that I do with the Western blot. I hybridize with primary antibody, wash it off, hybridize with secondary antibody, and this time the secondary antibody does not have an enzyme, but it has a dye that can be viewed in fluorescent microscope. And then I can also stain the nuclei with stains such as Hoist or propidium iodide, and then I can image using a fluorescent microscope. And this is what you see: the stained nuclei, and then you have protein that is stained right here, green. You can actually use more than one primary antibody. And more than one secondary antibody. You could use one primary antibody that has a dye that can be interacted with a secondary antibody with a red dye conjugated to it, and you can use another primary antibody that can interact with a secondary antibody with a green fluorescent dye that is conjugated to it. And so you can after you hybridize with primary antibodies, and then you hybridize with secondary antibodies, and then you stain the nuclei and you mount the images, and you can look at them under microscope, fluorescent microscope. Not only will you see the quantity of the expression of each proteins, but which location they're expressed at. They can be expressed in the cytosol or on the plasma membrane right here. You can see that the nuclei, and surrounded by the nuclei are the green labeled proteins, and then you see this cytoskeleton protein that is expressed mostly in the cytosol and toward the plasma membrane. So these are some of the really basic techniques that are used to isolate and to detect protein expressions in cells.